Welcome to Lakshman Biology. Till now, we have seen about evidences from paleontology. Second evidence for evolution is embryological support for evolution or embryological evidences of evolution. First of all, if you see the embryology means study of early development of an animal or organism. Study of early development. Study of early development of an animal is called as embryology. Ernest Haeckel is called as father of embryology. Father of embryology. And even he given embryological support of evolution. Embryological support. Embryological support of evolution given by Ernest Haeckel. How he given embryological support of evolution? What he did, he observed the early developments or embryology. He observed the embryology or early development stages, embryology of vertebrates. Vertebrates means like fish, amphibians, reptiles, aves, mammals, etc. He observed the early developmental stages of vertebrates. After the observation, he found that what he found? He found that there is some characters which are same in all vertebrate embryos, but that characters are disappear in adult stages. Here found that some characters same in all vertebrate embryos. Example, if you see the one of the example for that character, but those should differ that but that show difference in adult stages, shows differences in adults. Based on that, we can say that animals which has similar characters in embryo has the common ancestor. Those are evolved from the common ancestor. It will give evidence for the common ancestor. If you observe what he observed, one example for the observation of Ernest Haeckel, he observed the embryos of all vertebrates. Example, this is the embryo of fish. Embryo of fish. And here it is the embryo of amphibia. Embryo of amphibia. And amphibia means frog. It is the embryo of frog. Then this is the embryo of reptiles. Embryo of reptiles. This is the embryo of bird embryo. This is the embryo of bird. It is a reptile and this is bird. And here it is embryo of human being, human embryo of human. If you observe the embryo of fish, all are vertebrate embryo only. Embryo of fish contain gill slits. Embryo of frog contain gill slits. Embryo of reptile contain gill slits. Embryo of bird contain gill slits. Even embryo of human also contain gill slits. These gill slits are non-functional in embryo. Hence, it is called as vestigial gill slits. Vestigial gill slits. Vestigial gill slits are present behind the head in the all vertebrates. But later, if you see the adult, only in the fish adult stages, the gills are becoming to functional. Gills are present even in adult fish. Remaining frog not contain gills in adult stage, reptiles not contain gills in adult stage, birds not contain gills in adult stage, even human being also not contain gills in adult stage. But in the at least few stages in embryonic stage, all contain same character. Hence, Based on that, we can say that these are contain similar ancestor. There is one scientist, name of the scientist is Carl Ernest von Baer. What he did, he disproved, which he disproved? He disproved the embryological evidence of Haeckel. Embryological evidence of evolution is disproved by Ernest von Baer. How he disproved? How he disproved? He noticed what he noticed. He noted that embryos never pass through adult stages of other animals. Embryos never pass through adult stages of other animals means, example, this is the embryo of fish. Embryo of fish. Embryo of fish not become into human, adult human. Adult human is not formed from embryo of fish. Embryo of fish become into adult fish. Embryo of human become into adult human. Okay, if all embryos are same, according to Haeckel, all embryos are same because all contain similar feature. But according to Van Baer, embryos are different. Okay, hence embryos are different. How he proved that embryos not contain the same features, or embryos not show the similarities based on the observation? That is, he told that embryos never pass. He noted that embryos never pass through adult stages of other animals. Embryo of one animal not become into another animal. Then there is some examples for embryological support of evolution. First one is similarity in early development. If you observe the every animal, every vertebrate mainly, the sperm joins with the ovum leads to formation of zygote. Zygote in all animals. Zygote become into morula. Zygote start cleavages and form morula. Morula become into blastocyst. Blastocyst. Blastocyst again become into gastrula. Three germinates are formed in gastrula. Those things happen in all animals. These events are same in all animals. That is also will give evidence for the embryology. Next, we will see about biogenetic law or recapitulation theory. Biogenetic law or recapitulation theory. Biogenetic law recapitulation theory also one example for the evidence from the embryology. At the starting, what happened? Von Baer, scientist, he is also called as the father of modern embryology. Father of embryology is Ernest Haeckel. Father of modern embryology is Von Baer. He proposed one law that is called as Baer's law. What is the Baer's law? According to this Von Baer law, during embryonic development, the generalized features like brain, spinal cord, axial skeleton, aortic arches, everything are common in all vertebrates. Okay, at the starting, brain, same type of brain is formed in fish, reptile, aves, mammals. Same type of spinal cord is formed, same type of axial skeleton is formed. Axial skeleton means skull, vertebral column, ribs, etc. Same type of aortic arches, those are blood vessels, are formed in vertebrates. Okay, that is the one way. Means embryos, all embryos contain certain characters that are same in all embryos during the development. Later, there is another scientist. Name of the scientist is Haeckel. He modified the Von Baer's law into the biogenetic law. Biogenetic law, hence biogenetic law is proposed by Ernest Haeckel. Ernest Haeckel also given the evidences for the embryological evidence of evolution. That Ernest Haeckel modified the Baer's law into biogenetic law. What is the biogenetic law? Haeckel biogenetic law explained that antogeny repeats the phylogeny. Antogeny repeats the phylogeny means present day organisms life cycle, any stage of life cycle in present day organisms exhibit what it exhibit its ancestor character. Meaning of that is the present day animal, any one stage of present day animal exhibit the its ancestor character. One example for the ontogeny repeats the phylogeny or theory of biogenetic law, tadpole larva. If you observe the tadpole larva is the larva of frog. Later what happened? Tadpole larva is become into adult frog. This is the frog. Tadpole larva become into frog. But here tadpole larva not during, if you see the excretory products of tadpole larva, it is similar to fish, not similar to frog. Adult frog will excrete the urea, but tadpole larva will excrete the ammonia. Based on that, we can understand that tadpole is excreting ammonia. Fish is excrete the ammonia. Fish character. Amphibians are found from fishes. Fishes. Fish character is exhibited by life cycle of amphibia. Anyone life cycle of amphibia. Hence, it is an example for biogenetic law, which will indicate the origin of amphibians occurs through fishes. Next, another example for the embryonic evidence, embryonic evidence of evolution. During the development of heart in the higher vertebrates, example, if you see the mammals contain four-chambered heart. That four chambers will not form at the same time. First, only two chambers are formed in human heart in embryonic development. After that, three chambers are formed in embryonic heart. After that, we can see the formation of four chambers. Two-chambered heart we can observe in the fishes. First fish character we can see in the human heart. First, only two chambers are there in embryonic stage. Later, become into three chambers. It is the amphibian character. Later, we can see the formation of the amphibian or reptile, both three-chambered heart. Finally, we can see the formation of four-chambered heart. That is also here embryonic stage will be recapitulate its ancestor. Hence, it is also embryological evidence and it is also an example for biogenetic law. Next, another example, resemblance among invertebrate larvae. If you see the larval form embryos, the embryonic larvae of different animals are same. Example, if you see the Annelida and Mollusca, both are different phylums. But larva, both contain one similar larva called as trochophore larva. It is also will give evidence for the embryology. Echinoderms, hemichordates. These are two different phylums. Echinoderms and hemichordates both has similar larva. Next, the larva resemblance points of common ancestor. Meaning of that, the larva of one animal show the characters of its ancestor. Example, ammoset larva. Ammoset larva is larva of lamprey. It is the petromyzon. Lamprey also called as petromyzon. It is a cyclostome. Means cyclostomes means that jale species. The lamprey larva is ammoset, but it will exhibit the character of amphioxus. Amphioxus is separate group. It is also called as brachiostoma. It is a cephalochordata animal. Cephalochordata. But here, cephalochordata is different, cyclostome is different. Larva of cyclostome will exhibit the cephalophore adult character. That is also an example for embryological evidence. Next, degenerate organisms. Degenerate. That is also called as retrogressive metamorphosis. In few animals like Saculina tunicates. Saculina tunicates, example for tunicate is Headmania. These are some groups of animals that will show the retrogressive metamorphosis. Retrogressive metamorphosis means larva contain many organs. Well developed, larva is well developed, which contain many organs, but in adult stage, many organs are degenerate. That is only called as retrogressive metamorphosis. Even the tunicates or Headmania contain notochord in larval stage, that is later become into degenerate. But larval forms of these all these animals, tunicates, saculina, those will show the similarities. But adult stage, all features are become into degenerate. Next, temporary embryonic structure. It is also will give evidence for evolution. If you see the bird embryo, in embryonic stage, this is the bird. There is a beak beak poses or beak of bird contain some teeth like structures. Some teeth like structures you can see in the beak of bird only in embryonic stage. Later it will become into disappear. There is no use of these teeth like structures in the embryo, or simply you can say the teeth buds like that. But it should be helps in the adult, but adult not contain, it is disappear in adult. That is also will give one evidence for embryology, evidence from embryology. Thank you.